Inheritance and variation, chapter number 3rd. In that today we'll discuss about crossing over. Previous lecture we discussed about linkage, those genes which are present on the same chromosomes that inherit together, that is called linkage. But here the crossing over, that means the separation of linked gene. The separation of linked gene is called the crossing over. ज्या chromosomes वर जे जीन असतात ते सेपरेट होतात कशामुळे बाय क्रॉसिंग ओव्हर and that phenomenon is called the crossing over, and this crossing over which is discovered by Morgan, and Morgan said on that crossing over, that is the separation of linked gene. That means here genes that is A and B, these B genes which are separate by the crossing over. When the crossing over takes place on that gene, and this gene here genes are transferred that chromosomes, and these chromosomes genes are transferred from that chromosome, that is the segment are separated from the crossing over. That phenomenon is called the crossing over. Here one of the definition, that is it is a process. ही एक प्रकारची प्रोसेस आहे. It is a process that produce new combination of gene. Why new combination of gene? Because of genes are crossed over. That is the new combination or recombination of gene by interchanging of and exchanging of corresponding segment between non-sister chromatids of homologous chromosomes. Here the homologous chromosome here, this is the one chromosome, this is another chromosomes, and these are the homologous chromosome. One is maternal and one is paternal. These chromosomes are the homologous chromosomes here, maternal and paternal. One is mother chromosomes, one is father chromosomes. These chromosomes, when exchange or gene by exchanging the corresponding segment between non-sister chromatids here. Non-sister chromatids means here in the same chromosomes, these chromatids are called the sister chromatids, same chromosomes. But the homologous chromosomes, these chromosomes are called the non-sister chromatids here. In that non-sister chromatids, the crossing over takes place. Non-sister chromatids crossing over takes place, but the word is here the corresponding segment between non-sister chromatids. Corresponding segment means here the segment of chromatids, those chromatids which are close to each other. Those chromatids which are close to each other, that means crossing over takes place in these section. These chromatids crossing over takes place segment is come here. That means here this is one of the crossing over takes place and it is visible in the diplotin stage here. When crossing over takes place in the packaging stage, Chayes meta point, that point is called a Chayes meta point. Crossing over is when visible. When visible, it is visible in diplotin stage. It is visible when question asks when crossing over is visible. It is visible in diplotin stage. This is the crossing over. What is the crossing over? It is the process that produce new combination of gene. That produce new combination of gene here by exchanging or corresponding segment between non-sister chromatids of homologous chromosomes, and the T.H. Morgan says that the crossing over is the separation of linked gene. What Morgan says, the separation of linked gene is called the crossing over. It is the simple definition of crossing over. And then next another definition, that is it is the exchange of the chromosomal segment. It is the exchange of the chromosomal segment between non-sister chromatids of homologous chromosomes. It is the exchange of the chromosomal segment between non-sister chromatids of homologous chromosomes is called the crossing over. And one main thing is formed because of this crossing over, that is the recombination or new combination of gene. That main thing which is formed, that is the recombination or new combination of gene. एक नवीन काहीतरी व्हेरिएशन निर्माण होते, that is the new combination or recombination. This is the crossing over. Then we see the Morgan's experiment. And Morgan's experiment is based on or show the linkage and the crossover or linkage and crossing over. Morgan's which born 1866 and died 1945, and he was the biologist, and he used for his experiment Drosophila melanogaster. Drosophila melanogaster, which is the fruit fly, which Drosophila, why he used for his experiment Drosophila melanogaster, because his lifespan very short lifespan, at least 2 weeks, and it has the high rate of reproduction. It has high rate of reproduction. खूप संख्येने ते तयार होतात, and lifespan त्यांचा कमी आहे, and at least 2 weeks. That's why he used, because of the much more conclusion or much more result to see the easily or earlier. That's why he used the Drosophila melanogaster, that is called the fruit fly. Here he discovered what he discovered. He discovered the principle of linkage. He discovered the sex linkage. He discovered the crossing over here, and for his key work, he plays key work in the genetics, and for his work that he awarded the Nobel Prize in 1933. He awarded the Nobel Prize in 1933 in physiology and medicine here. And his experiment Drosophila melanogaster, he done the linkage, crossing over first, and he done the linkage or show the linkage with experiment cross first, and he show the crossing over with his experiment cross second. He used the dihybrid cross of Mendelian cross like dihybrid cross. He used the dihybrid cross, म्हणजे त्यांनी 2 कॅरेक्टर निवडले Drosophila melanogaster मधले dihybrid cross सारखे as like of the Mendelian dihybrid cross, but the ratio of dihybrid cross of the Mendelian, that is the F2 generation ratio here, that is female is yellow bodied and white eyed, and male brown bodied and red eyed. Male here. Here female, which is the recessive here, Drosophila melanogaster. Female here, which is recessive, that is the sex linked gene here, and male, which is the dominant, show the dominant character, that is wild type, giving here in book here, say that the wild type here, wild type. Yellow, that is Y, because of the recessive trait, that is the small y, and white, small w. Here two genes which are the recessive gene, and brown bodies and red eyed, because of the show the dominancy here male, that means here giving not a capital here but giving plus here, that is y plus and w plus here. That is the wild type of the male, which is y plus and w plus. Here giving not a capital, capital घेतलेला नाही ह्याच्यामध्ये, प्लस कॅपिटलच्या ऐवजी प्लस घेतलेला आहे, and it is brown bodies and red eyed and recessive, that is the yellow body and white eyed. This is the female and this is the male. When this is crossed with each other, this is Morgan said named that the cross first. Cross first. And what cross first here? Parental generation. Parental generation. That is parental generation female. That is the recessive female here. That is the sex linked chromosomes here. That is the sex chromosomes here. This is the two sex chromosome, that means XX. Here the female. And cross with male. Male Drosophila melanogaster here. This is the male, which show the one say X chromosomes and one is Y chromosomes here. This is the male. Here the gene that is yellow bodied and white eye here, that is the small I, small w, small y, small w here. And this is the wild type, which is show the dominant here, y plus w plus. One thing is here that those genes which are present on the X chromosome, those genes which are not present in the male on the Y chromosomes. Because of this show this type of chromosome, that is the X and Y chromosome. Y chromosome not show any gene here. The dominant gene is only present on the X chromosomes of Drosophila melanogaster here. This is yellow and white. And given book here the wild type, or it is dominant, and it is the name, that is the brown bodies, that is the brown and red, or wild type. When this cross with each other, when this cross with each other, in the book there are not giving the gamete here. The book in the SCRT also and the book in the NCRT not giving the gametes. That's why the confusion is here. One confusion is here, not giving here or not right here brown and red, and second confusion here, not giving the gamete here. Now we see first the gamete. Gamete, when gamete is formed, here the same types of chromosomes here, the same types, that is the one type of gamete is formed. This circular structure is the centromere here. The circular structure is the centromere. This is the gene, that is the Y and W. These are closely related. हे जवळ जवळ gene आहे तर this is the closely related gene. This is the same type of gamete here, but here the two types of gamete are produced here, Y plus and W plus, and another gamete here, that is Y chromosome here. This is the gamete. When these gametes are fused, the F1 progeny will be produced. Here these gametes are fused, that means here because of the dominancy we also write here Y plus W plus and Y W, and this fuse with this, that is this is the female and this is the male. This is the F1 generation here. This is the F1 generation, which is, this is the wild type. That means brown bodies and red eyed. Why wild type is called here? Because of the dominant gene are present here. That's why brown bodies and red eyed. This is the wild type. And this is the yellow and white. When these F1 generation are selfing with each other, then we see the what type of F2 generation will be produced. When these F1 generations selfing with each other, that is the first that gametes are formed here. Gametes are formed here. Here female show the four types of gametes here. The first gametes is as it is, then the second gametes is this as it is, then when meiosis takes place, because of crossing over, if crossing over takes place here not the actual crossing over because these genes are closely related, but when crossing over takes place, that means the low chances of crossing over. When that crossing over takes place, here the two new types of gametes are formed here, that is the Y plus W and Y W plus here. The new types of gametes are formed. That is the four types of gametes are formed from the female and male. There are two types of gametes are formed. When these gametes are fused with each other, that F2 generation will be produced here. And F2 generation, when these male, that is two gametes, we know here yellow and white body. Here two gametes here, male show the two gametes here and female show here the four types of gametes here. The first gamete is Y plus W plus, that is the dominant. The second gamete is YW. The third gamete is Y plus W, and the fourth gamete is YW plus. When these gametes are fused with each other, then what will be the progeny here? When these gametes fused with these gamete, here Y plus W plus, Then when these gamete fused with this, that is Y plus W plus then this gamete fused with this, that is YW, YW. When this gamete fused with this, YW and Y chromosomes here. Then this gamete fused with this, one Y plus only W here. When this gamete fused with this, Y plus W, then this fused with here the first paragraph, first fusion of these gamete, it shows because of the dominancy, brown bodies and red. Here also brown and red. Here not any dominant character, they are yellow and white. Here also yellow and white. But here one is dominant here, that is the brown and white. Here also one dominant, brown and white. Here one dominant, that is the red, that is yellow and red. This is also yellow and red here. This column all are daughters here. These are all daughters. And these are all our sons here. And here when he see the ratio of that new combination is not takes place, very rare here, the parental types are more here. He see that in experiment he saw the more parental types are produced in the F2 generation. त्याला फक्त तिथं नवीन कॅरेक्टर्स असणारे, नवीन व्हरायटी असणारे त्याला फक्त कमी दिसून आले. And that type, that is the parental type, he saw that 98.7% and new recombinant type only 1.3%. That means the conclusion is that experiment here, that gene Y and W are tightly linked. The conclusion is that Y and W gene are tightly linked. And because of tightly linked genes show the less recombination, and tightly linked gene show less recombination. This is the first cross of Morgan's experiment and used the Drosophila melanogaster, and in that the parental generation show the 98.7% and the recombinant type show only 1.3% because of tightly linked gene, because of closely linked gene. Then the next cross, here the second cross here which show the crossing over here. First cross show the linkage, but second cross show the crossing over here. In that cross here, that is giving here two characters, that is the eye color and wing size. We know that eye color, that is the white and red, that is the eye color, and wing size, that is the normal and miniature here. That female which is show the white and miniature characters here. It is the recessive character, that is the white, and miniature, that is the M. Allele show the white, that is the W, and miniature show the M here. Then these females and the male show the red and normal here. Male show the red, that is red which is the dominant trait over the white, that is the W plus, and normal show the dominant over the miniature, that is the M plus here. This is the dominant trait and this is the male. Y chromosome not show any gene of that trait here. Here the gametes are formed. Gametes are formed here. Same types of gametes are formed. That is the white and miniature. And here show the gene, that is the white and miniature. Here but show the two types of gametes here, that is the X and Y. Also here, that is the dominant, here show the W plus and the M plus here. When these gametes are fused with each other, when these gametes are fused with this gamete, that show, that is normal and red and normal daughter here. Red and normal daughter. Why it is red? Because W plus here over the dominant, over the W only. That's why red, and normal, that is the M plus which is dominant over the M here. That's why red and normal. But red and normal daughter, but the son is white and miniature. White W and the M which is the miniature. When these F1 generation, this is the F1 generation, when these F1 generation will selfing with each other, when these F1 generation selfing with each other, first of all the gametes are formed, then the F2 generation will be produced. This is the second cross second, and F1 generation, that F1 generation which show the red and normal. What red? Because of this is the dominant, that is the W plus, normal M plus here, and the male show, that is the white and miniature here. When this crossed, there are female produce the four types of gametes here. That gametes, this are as like it is. That means a W and M here. Then second gamete, that is the W plus and M. This circle-like structure which is the chromatin here. Centromere here. And then the third types of gametes which is the W plus here and M here. And four types of gamete here which is W and M plus here. The four types of gametes which are produced, and here male which produce the two types of gametes here. One is the W and M and second is the Y chromosome. It is the two types of gametes here. When these gametes are fused, when male gametes fused with female gametes here, this is the male gamete, there are two male gametes, and female gametes, there are four female gametes here. These are the fusion of these gametes. Here the first fusion, that is the W and M, normal. This is also red and normal. This is red and miniature. Because of red, which is the dominant, red and miniature. This is also red and miniature. This is white and normal. This is also white and normal. This is all are the daughters and this is the sons. Here the new recombination are produced in much more quantity because of the crossing over takes place. Because of the genes are located some distance away in the chromosomes here. Gene from eye color and wing size which are loosely arranged and the recombination which is a parental type. Parental type which is 62 point here, 62.8% here. Parental type which is parental types are produced. Parent सारखे मॉर्गन्स ला काय दिसले 62.8% एवढे ड्रोसोफिला दिसले. त्याच्यानंतर रिकॉम्बिनेशन टाइप which is 37.2% here. And because of this parental and the recombination, here recombination are the much more from the crossover. Crossover पेक्षा recombination types हे इथं जास्त आलेले आहेत. And because of this, these genes are loosely. The conclusion is that the genes M and W are loosely arranged and show the more recombination here. What is the conclusion? That the gene W and M are loosely arranged and show the more recombination. Show more recombination. This is the Morgan's experiment. We see today cross first, cross second, and previous then we see what is crossing over. Then Morgan's experiment. He used that experiment Drosophila melanogaster and these two crosses. First cross is the cross of the linkage cross. Show the linkage. Linkage दाखवतो. Linkage मुळे recombination जे आलेलं आहे ते कमी आलेलं आहे. That is 1.3% only. आणि सेकंड क्रॉस जो आहे तो काय दाखवतो आहे? तो क्रॉसिंग ओव्हर दाखवतो आहे. त्याच्यामुळे रिकॉम्बिनेशन किती आलेलं आहे? That is 37.2% here. This is Morgan's experiment. I hope you all understand. Thank you.